Welcome to Study Loop.
We are being asked to determine what would happen to the repressor in the lac operon if the binding of the repressor to lactose is deficient. So let's put that in context a little bit. Up here, up top, we have the lac operon, which is basically the genes so the genes LAC-Z, LAC-Y, and LAC-A, those are genes that make enzymes that can consume lactose, that can break down lactose. And the operon also has a CAP binding site, which acts as a promoter, and which binds to the CAP protein, which acts as a promoter, and it has an operator site to which the repressor bind, which represses the production of the enzymes that consume lactose. And the reason that the cell wants to control this is because if there's glucose, it would preferentially consume glucose because glucose is a simpler sugar, and it provides more efficient energy than lactose. So when there's glucose present, you don't want to be making lactose. When there's no glucose and there is lactose, then you want to go ahead and consume lactose. So the cell needs two factors it needs to be able to differentiate these two factors in order to turn the operon on. So the first case here, in low glucose and high lactose, you have the CAP protein on and you have the repressor. So the repressor is drawn on the bottom here as this brownish rectangle, but it's not uh, up top here where the RNA polymerase is binding, because if it was, then the RNA polymerase wouldn't be able to bind. So the two conditions for the lac operon to be on is that the glucose must be low and lactose must be high. How does the cell know that glucose is low and lactose is high? Well, glucose prevents the production of CAMP, which is cyclic adenosine monophosphate. Okay, CAMP binds to the CAP protein. The CAP protein stands for catabolite activator protein, I believe. So the CAMP binds to the CAP protein, and then that activates the CAP protein to bind to the DNA and promote transcription. So when glucose is low, there's not enough glucose to block the production of the CAMP. Levels of CAMP go up, binding of the CAP protein to the DNA goes up, levels of transcription go up. Lactose binds to the repressor. So lactose, when it binds to the repressor, it prevents the repressor from binding to the DNA. So if it prevents the repressor from binding to DNA, then the repressor is not on the DNA to stop transcription. So when lactose is present, it binds to the repressor. Repressor comes off of the DNA. So now the question is asking, what happens when you take away this interaction between the lactose and the repressor? So basically, the repressor is insensitive to the presence of lactose. Well, the repressor would constantly be on the operon, and it would prevent the production of the enzymes that break down lactose. Okay, so let's look at our answer choices now. Choice A is the repressor will bind to lactose. Well, that's wrong right away, because the question is saying that the thing that is being affected is the binding of the repressor to lactose. Choice B, the repressor will bind the operator in the presence of lactose. Yes, that is correct. When lactose is present, the repressor will be unresponsive to the presence of lactose and will not come off the DNA. Choice C, the repressor will not bind the operator in the presence of lactose. That is not true, because that would happen only if things were working correctly. Choice D, the repressor will not bind the operator, and that's the same reasoning as C, so choice B is the clear winner here.